Hi, I'm Dr. Greg Ketchum. I'm the principal of Talent Planet, a leadership development and media production firm. And I'm joined today by Richard Gabriel, who is the Chief Operating Officer and co-founder of GLG Pharma. Richard, welcome.
Thank you, Greg. It's very nice to be here with you.
We're happy to be here with you today too. Richard, I understand that you and your colleagues are working on a new and very promising targeted form of cancer therapy. What can you tell us about that?
Well, let's take ovarian cancer. 40% of the women survive the standard chemotherapy and no one knows why. Okay, that's number one. So take that number. The problem has always been in screening laboratory samples for patient recommendations. You know, a pathologist recommends to an oncologist what the patient should be doing is that the cells always change, and they're always changing because we're not culturing them correctly. So how cancer works, and particularly with ovarian, is it fools our immune system into thinking that everything's okay. So what we've done in the lab, simply, is to fool the cancer cells into thinking that they're still inside the body. And now we can culture them up and they don't warp. And the screening results we're going to get in combination drug therapies are going to be much better.
Wow. That sounds like very, very promising. How is this different, Richard, than what's come before in the diagnosis and treatment of cancer?
Well, there's only really two kinds of treatment. There were only a couple of companies doing what we would call drug screening, effective drug screening. One system is in what they call the PDX mouse model. So they insert the tumor in the mouse, they take the tumor from the patient, they insert it in the mouse, and it takes a relatively long time period to grow up. In the case of ovarian cancer, where a patient is in stage 3 or stage 4, even before they're diagnosed, 90% of the patients are in stage 3 or 4, meaning that we don't have a lot. So the PDX mouse model takes 36 to 48 weeks. We were looking for something that's faster, cheaper, easier to expand, and reliable.
Wow. So my understanding of what you all are doing is you say you found a way to fool the cancer cells, take them out of the patient's body, put them in, the lab petri dish or whatever, and try out different treatments on them so that the patient themselves don't have to go through so many different trials of different drugs. Is that a fair understanding of what you're doing?
Well, in an oncology environment where a patient comes in a particular ovarian cancer, where a patient does not have a particular marker that is identified with a new improved therapy, they go through standard therapy. So if we can, let's say that we can take those 40 patients and we can raise, instead of a 50-50 shot for those 40 patients out of 100, we can raise it to 90.
Yeah.
So that means that we then can say confidently to the oncologist that this particular patient has a 90% chance of surviving this very awful chemotherapy regime. What about the other 60 or 70? Well, that tells the oncologist maybe they should go into clinical trials.
Okay, so it sounds like it's just a way of, in a sense, tailoring the treatment more to the individual.
Yes, it's the only way you can change protocol in an oncology situation is you have to have data. And the only way to get data is you have to have reliable cell culturing technologies which don't exist.
Okay. Well, Richard, in my experience, most people get involved in things in life because of some personal meaning or some personal reason. Is there something like that for you in terms of your involvement with the diagnosis and treatment of cancer, particularly ovarian cancer?
Yeah, there is. Yeah, personally, yes. I don't generally like to talk about it because it's every family that's gone through a situation where a loved one has had cancer and it's not a good outcome, has the same feelings that I have about it. My first wife, when I was 31 and she was 29, she passed away from leukemia when our daughter was 3 years old. And it was a 3-year downward spiral. It destroyed the family. It destroyed just everything. I mean, it just is it's an awful experience. Everyone tried really hard, all the oncologists, the laboratory people, the nurses. I mean, it was just an awful, awful experience.
Wow. So sorry that you and your family had to go through that. That does sound like a really terrible experience for all involved. But, you know, it looks like some good has come from that with your involvement in doing this cutting edge research. Now, if our viewers want to get involved, how can they get involved, Richard?
Well, we're doing we're listed on a fundraising site, which is an internet portal called WeFunder. It is actually one of the first investment vehicles that was signed in by the Jobs Act several years ago, where ordinary individuals can invest in private equity companies prior to a listing on the NASDAQ. So before, you couldn't do it. So that's the site that we send everyone to. The investment can be as little as $100, and your money will be used to help us build and get these products into the market as soon as possible.
Wow. Okay. So that's wefunder.com/GLG Pharma.
That's correct. Yes.
Okay. Well, thank you, Richard, for taking the time to chat with me today. I've been speaking with Richard Gabriel, who is the Chief Operating Officer and co-founder of GLG Pharma, about some new and very promising targeted forms of cancer treatment and therapy. Richard, thank you so much.
Well, thank you very much, Dr. Greg. I really appreciate this opportunity. Thank you.